Okay. Okay. Very good evening, my dear friends. Regarding of this CPGET 2024 free online coaching. Today's our eminent resource person, the very most experienced subject expert. That is none of the other than Dr. Pamuru Manjula Madam Garu, Assistant Professor, Department of Botany, Food and Nutrition, Rbvrr Women's College, Autonomous, Hyderabad. Since Madam has given her services to our young aspirants, those who are attending CPGET Botany from 2020 onwards, that means from Corona pandemic situation onwards, Madam has rendering her valuable services to the young aspirants, those who are going to attend CPGET examination Botany. So we are very happy and thankful to our Dr. P.Manjula Madam Garu. Please welcome and share your screen and start your session, Madam. Please welcome. So remaining people will going to join. I have informed them. Please start your session, Madam. Okay, thank you very much, Madam. Please
Okay, thank you, sir.
okay, please click on slideshow.
Okay, sir. Thank you, sir, for giving this opportunity. So today's topic is internal structure of dicot and monocot root. So generally roots. So generally here the differences between the dicot, main difference between dicot root and monocot root is here the dicot roots are taproot, whereas the monocot roots are the fibrous root. So in dicots, that is taproot system is seen, whereas in monocots, the fibrous root system is seen. So dicots are the taproots and which consist of single primary root, from which secondary and tertiary roots will develop and grow vertically downwards through the soil. So generally roots, they are non-green part of the plant and that is present below the soil. And they do not have any nodes and internodes. So generally when we compare the stems, so stems have nodes and internodes. But here nodes and internodes are absent. And the roots in dicot plants, they are generally mostly similar in structure. But the length or the thickness of the root and complexity of the root system, it may be different. So generally in some dicot plants that might have, so here the roots are modified into different structures for the, and also the functions also for purposes like respiration. So it is called as the respiratory roots. So in some, the roots will store the food materials and storage roots. And another one is the mechanical support. So these are the different here, the modifications of roots. So a typical root which consists of different parts like root cap, meristematic zone, and zone of elongation and zone of maturation. So now we will discuss about the primary structure of dicot and monocot root. So the primary root of the dicot root system, it is the taproot, which grow vertically downwards to the towards the soil. So from the taproot, that is secondary roots will arise, so which might grow sidewards as well as the downward. So tertiary roots also, it might arise from the secondary roots in order to reach the greater depths and enable the absorption of water and minerals. So generally the main function of the roots is that is the absorption of water and minerals from the soil. So here the well-developed root system is generally seen in the root, in dicot root. So here the some differences, variations may be seen in when we compare to the hydrophytes and xerophytes. So generally in xerophytes, that is well-developed root system is seen. Whereas in hydrophytes, here the root system is not well-developed. So some variations may be seen. So here, tiny root hairs are also present in the dicot root system. So the dicot root, which can be both herbaceous and woody. So here the dicot may be herbaceous plant or it may be a woody or tree. So depending on the plant species. So the woody plant root system, it has the cambium, which enables the growth of the large plants with thick stem. So the anatomy or the internal structure, when we take the transverse section or cross section of the root of dicot or monocot, so it contains the following parts. So first one is the, it is called as the epidermis. So generally in roots, it is called as the piliferous layer or epiblema. So another term, that is generally for the roots, we use the term piliferous layer or epiblema in place of epidermis. Okay, so here the epidermis, that is the outermost layer, it is of the roots, so which is composed of compact layer of the thin-walled polygonal parenchymatous cells with no intercellular spaces. So generally epidermis, it is single layered and it is the outermost layer and it is made up of parenchymatous cells. So the cells shape may be polygonal in shape, okay, without intercellular spaces. So the epidermis of the root does not have the cuticle or stomata. So when we compare the stem, so here on the epidermis, cuticle is present and stomata is present in the stem. But here the cuticle and stomata are absent on the epidermis of the root. Okay, so some of the cells of the epidermis which gives rise to specialized root hairs that are present on the maturation zone of the both monocot and dicot root. So here the root hairs are present on the epidermis. So here generally in the stem and the epidermis trichomes are present, but here the root hairs. Okay, so due to the absence of intercellular spaces and presence of hair cells, the layer it is called as the piliferous layer. So the term piliferous means that is on the epidermis, here the root hairs are present, hence it is called as the piliferous layers. So this is this term piliferous layer or epiblema which is used for roots only. So in monocots, so the epidermis it is also called as a rhizodermis and which consists of hair forming trichoblast and non-root hair forming the atrichoblast. So here the trichoblast and atrichoblast. So a specialized multi-layered epidermis is present in the monocot plants. Like, so here some variations may be seen. So in the orchids, so which is called as the velamin roots. So the velamin is involved in gaseous exchange. So here in orchids, that is the specialized multi-layered epidermis is seen in the orchids. That is velamin roots are seen in the orchids. So it mainly helps in the exchange of gases. So generally orchidaceae members, that is, they are epiphytic. They grow on the other plants. So here they absorb the moisture from the atmosphere. So that's why here the velamin roots are seen. So the epidermis is short-lived and in older roots, which is replaced by the lignified suberonized exodermis. So epidermis, which is replaced by the exodermis. So this exodermis, which develops from the outermost cortical cells that form an additional Casparian band. So generally epidermis is present, but here it is replaced by the exodermis, so which is developed from the outermost layer of the cortex. So in dicot roots, the epidermis along with the other tissues of the roots is peeled off and later replaced by the cork cambium. So in shoot-borne roots of the monocot plant, so the epidermis which can persist and form the protective cuticle. So epidermis in both types of roots which involved in providing the protection. The main function of here the epidermis is protection of the internal tissue and the minute hair root hairs which enable the absorption of water and minerals from the soil by providing the large surface area. The main function of the epidermis is the protection. Okay. So this is about the internal, that is the epidermis of dicot and monocot. So next one is the cortex. So cortex, it is the tissue which is present beneath the epidermis and which is composed of many layers of cortical cells. So the cortical cells in monocots are thin-walled, multi-layered parenchymatous cells that have efficient. So the cortical cells in the monocots are thin-walled, multi-layered parenchymatous cells and that have sufficiently large intercellular spaces between them. So the cortex of dicot roots, however, consist of sclerenchyma in the addition to the parenchyma. So here the cortical cells in monocots are thin-walled and multi-layered and they are made up of parenchymatous cells with large intercellular spaces. So whereas in cortex of the dicot roots, they are however consist of sclerenchyma in addition to the parenchyma. So here both sclerenchyma is parenchyma is present in the cortex of the dicot roots, whereas in monocots only it is made up of parenchymatous cells. So that is the difference. So as the epidermal cells, the cells of the cortex are also non-photosynthetic as they do not have chlorophyll. So some dicot plants like Tinospora and Trapa have the chlorophyll in the cells of the cortex and are photosynthetic. So generally, and the roots, that is generally the roots are present inside the soil. So here the chlorophyll is absent, but some plants like Tinospora and Trapa, that is in the roots also chlorophyll is present and they helps in the photosynthesis. So in some plants, the cells of the outer layer of the cortex undergo suberization and form the single layered or multi-layered exodermis. So the cells of this region which store the starch in the form of starch grains and contain the leucoplast. So the most important function of the cortex is the movement of water from the epidermis to the inner tissues. The region of the cortex is wide in monocot roots when compared to the cortex of the dicot roots. As monocot roots have as many as 18 layers of parenchymatous cells. So here it is in monocots, the cortex has many layered parenchymatous cells. So next one is the endodermis. So the endodermis, it is the another layer of the dermis tissue, that is dermal tissue. So first epidermis or epiblema or piliferous layer, which is followed by the cortex. Next one is the endodermis. So it is present between the cortex and the central vascular tissue of the root. And here the endodermis act as a barrier between the two layers. So it is composed of tightly packed barrel-shaped cells and that lack any intercellular spaces. So it is single-layered and tightly arranged cell without intercellular spaces. So the young cells of the endodermis, which possess internal strip of suberin and lignin, which result in the layer of Casparian strip. So this is also a special character which is seen in the roots. So Casparian strip. So as the cells mature, the strip becomes indistinguishable due to the thickening of the cells. So the internal layer of the cells of this endodermis, which gives rise to the adventitious roots in the monocot plants. So in the monocot plants, from this endodermis, that is the adventitious roots will be developing. Internal layer of this endodermis gives rise to adventitious roots. The young cells, that is lie opposite to the protoxylem, groups of the vascular bundle. So generally, these young cells which lie opposite to the protoxylem of the vascular bundle, and these cells are also termed as a passage cell. So another term, that is a passage cells, also seen in the roots. It is absent in stem. So passage cells are transfusion cells are present and they mainly involved in the conduction of fluids inwardly, that is from the cortex, and outwardly from the vascular bundles. So the number of passage cells which is equal to the number of protoxylem cells. So here first one is the Casparian strip. So Casparian strip, which is made up of suberin and lignin in the young cells of the endodermis. Okay. Next one is the internal layer of the cells of these endodermis, which gives rise to the adventitious roots. Next one is the cells which are lie opposite to the protoxylem. They are called as the passage cells, and they are young cells, and they are called as the passage cells. So the another name for this passage cells is the transfusion cells, and they mainly involved in the conduction of fluids inwardly from the cortex to, they observe from the cortex to from the cortex and outwardly from the vascular bundles. So the number of passage cells, which is equal to the number of protoxylem. So the number of passage, which is, that is they have, for example, here the number of protoxylem cells are 10. So the number of passage cells also 10. So thickened or older cells of the endodermis, they are also involved in the passage of fluids with the help of their plasmodesmata. So the main function here, mainly the roots function is that is a transport of water and nutrients from the soil to the all parts. It is applied to the all parts of the plant. So the endodermis, which regulate the flow of fluid between the cortex and vascular tissue and act as a biological checkpoint. And the next one is a pericycle. So pericycle, it is also single layered structure which is present underneath the endodermis and which is the most distinct layer of the cells between the dicot and monocot roots. So the pericycle of monocots, which is composed of single layer of sclerenchymatous cells with few parenchymatous cells. And the young cells of this layer are thin-walled but later become thick-walled due to the deposition of various substances. Whereas in pericycle of the dicot roots, which is composed of a type of the parenchyma called as a prosenchyma, which is defined by the abundance of protoplasm. They contain more amount of protoplasm. So in monocots, the pericycle can be, so here in dicots, that is in monocots, that is the pericycle is composed of sclerenchymatous cells with few parenchymatous cells, whereas in pericycle of dicot roots it is made up of parenchyma cells only. Okay, the main difference is here the pericycle in the dicots is the parenchymatous, whereas the pericycle in the monocots is the sclerenchymatous cells with few number of parenchymatous cells. So in monocots, the pericycle can be either uniseriate or single layered. As generally single layered in mage. So typical example for monocot root is the mage. So are multi-seriate or multi-layered. Sometimes multi-seriate, many layered pericycle, it can be observed in the smilax. So in dicots, lateral roots which are originated from the part of the pericycle present opposite to the protoxylem. So here in the dicot, lateral roots which develop from the pericycle, which are present opposite to the protoxylem. Thus, the lateral roots of the dicots are endogenous in origin. They are developing from the pericycle. Okay, so pericycle in dicot roots is involved in the formation of vascular cambium. And carp cambium is also formed in the dicot plants. So during the secondary growth. So the cambium is also not formed in monocot roots. So pericycle in dicots, which are also involved in the formation of vascular cambium and carp cambium. So generally in the dicots, secondary growth is seen, whereas in monocots, secondary growth is absent. So here the cambium is also not formed in the monocot. So cambium is also absent in monocot roots. So the pericycle, which is the important part of the root tissue, and as it is involved in the formation of lateral roots, cambium, and also provide the support to the vascular tissue present beneath it. So the main function of this pericycle is in the dicots mainly. So it mainly helps in the formation of lateral roots, cambium, and also provide the support to the vascular system. So next one is the vascular bundle. So the vascular bundles form the innermost tissue of the plant root and consist of alternate xylem and phloem units. So the number of vascular bundles which differ in dicots and monocot root. So here also difference is seen in the dicot root and monocot root. So generally in dicots, the vascular bundles are radial and exarch, and the number of such bundles varies between 2 to 6. Only in dicots, that is 2 to 6 number of vascular bundles are present. That is diarch to exarch. Okay, if 2 vascular bundles are present, so it is called as diarch. Triarch means 3 vascular bundles. Tetrarch, 4 vascular bundles. Pentarch, 5, 6 exarch. So minimum 2 to 5 vascular bundles are seen in the dicot. So in some plants like ficus, that is a polyarch condition, that is many vascular bundles are seen in the ficus. So some exceptional cases are there in that condition. So many vascular. So this point is also important. Generally in dicot, that is 2 to 6, that is exarch is the exarch condition, but polyarch condition is also seen in the ficus. So this is an exceptional case. That is here the more than 6 vascular bundles are seen in the ficus. So here the vascular bundles are here these vascular bundles are radial and exarch. Exarch means that is the protoxylem towards outside and metaxylem towards the inside. So in monocots, the vascular bundles are radial and exarch here also, but the number of vascular bundles is always greater than the 6. That is here the polyarch condition is seen. So in mage, Mage route, that is 20 to 30 vascular bundles are present, whereas more than 100 bundles also observed in the pandanus and palms. So here the variation. So more number of vascular bundles we can observe in monocots, that is from the minimum, that is 20 to 30 to 100 bundles may be observed in the pandanus and palms. So in both types of routes, the vascular bundles are arranged in the form of the ring around the central pith. So the vascular bundles, they are also termed as the radial bundles due to the alternate arrangement of xylem and phloem. So the xylem bundle in both monocots and dicots are exarch, that is protoxylem towards outside and metaxylem towards inside towards the pith, and protoxylem towards the pericycle. So the protoxylem lies towards the outside and metaxylem lies towards the center. So this condition, it is called as the exarch condition, is seen in the roots. So the xylem in the monocot is composed of oval vessels and xylem parenchyma, whereas the dicot is composed of polygonal and thick-walled cells. So here also variation is seen, difference. So in monocots, it is composed of vessels are oval in shape with xylem parenchyma. But here in dicots, it is polygonal and thick-walled. So generally xylem parenchyma means it is thin-walled, but here the thick-walled cells are seen. In dicots, the protoxylem vessels bear annular thickenings, whereas the metaxylem vessels have the reticulate thickenings. And here the xylem parenchyma and fibers are also absent in the dicot roots. So in dicot roots, this xylem parenchyma and xylem fibers are absent. So next one is the phloem. So the phloem bundles in dicots are present close to the pericycle and consist of sieve tubes, companion cells, and phloem parenchyma. And here phloem fibers are absent. So the phloem, it is divided into metaphlloem and protophlloem. So but they are not easily distinguishable. So the phloem bundles in monocots are present closer to the pith and consist of similar structures and cells. So xylem tissue in roots is involved in the conduction of water through the roots, whereas the phloem bundles are involved in the conduction of storage of food. So here the function of xylem is, that is the conduction of water, whereas the function of the phloem is the conduction of food materials. So next one is the conjunctive tissue. So the conjunctive tissue, so it is also seen in the roots. So the conjunctive tissue, these are the masses of parenchyma or sclerenchymatous cells that are present between the xylem and phloem bundles in the vascular tissue. So the amount of conjunctive tissue is more in monocot roots due to the large number of vascular bundles when compared to the dicot roots. So this conjunctive tissue is nothing but it is a parenchymatous or sclerenchymatous cell which are present between the xylem and phloem. It is called as a conjunctive tissue. Okay. So conjunctive, more amount of conjunctive tissue is seen in the monocot roots because more number of vascular bundles are present. So here the more amount of conjunctive tissue is seen. So in dicot plants, the conjunctive tissue is together with the pericycle and gives rise to the vascular cambium, whereas that is from the conjunctive tissue, here the formation of vascular cambium will takes place during the secondary growth. So no cambium formation occur in the monocot roots. Generally in monocots, secondary growth is absent. So these tissues are involved in the storage of food and also provide mechanical support to the root. So next one is the pith. So pith is the central mass of the tissue and composed of thin-walled parenchymatous cells in the vascular system of the root. So in dicots, the pith is less prominent or less developed. In some cases, it may be completely absent. In monocots, the pith is prominent with many cells and that are either rounded or polygonal. So the cells of the pith are generally loosely attached with large intercellular spaces in between. So the cells of the pith, which stores the food and help in the dispersal of air between the vascular bundles. Next one is the passage cells. So passage cells are the distinguished cells of the endodermis and that are involved in the conduction of water and other materials between the cortex and the vascular bundle. So here passage cells which are present in the endodermis region and they are mainly helps in the conduction of water and nutrients. between the cortex and the vascular bundle. So the passage cells are prominent in monocot roots, but are completely absent in dicot. So these are also called as the transfusion cells. So the passage cells of the endodermis are younger cells of the layer and they do not have deposits of the suberin or lignin. That is, the Casparian strips are absent in the passage cells. So the passage cells are present near the protoxylem and enable the radial flow of materials through the root system. So this is about the internal structure of the dicot and monocot root. So functions of the root. So the plants grow in marshy areas have roots and that come out to the soil surface in order to obtain the oxygen. So these roots are termed as a pneumatophores. So here some variations in the roots are seen. So pneumatophores mainly helps in the respiratory nematodes, which are mainly involved in the gases exchange. So in many dicot roots exist in symbiotic relationship with the microorganisms like fungi. So they are called as the that play essential role in the nitrogen fixation. Next one is the roots of some plants are involved in the propagation and also the dispersal of the plants. So these are the functions of the roots. So next one is the, so here this is the structure of the dicot and monocot root. So here the epidermis or epiblema are piliferous layer, and on the epidermis here the root hairs are present. So next one is the cortex. So next one is here the cortex, it is many layered, and here it is followed by the endodermis. In monocots, endodermis is single layered. So here these are the vascular bundles. So here the phloem, which is alternate with the xylem. Okay, so this yellow colored one, this is the phloem, and this red one is the xylem. So here the metaxylem towards the center and protoxylem towards the periphery. So central pit. So this is the internal structure. So we can observe by counting the number of vascular bundles, we can identify the dicot root or monocot root. So here many vascular bundles are present, more than 6. So it is a monocot root. Here only 4 vascular bundles. That is 4 vascular. So tetrarch condition. So in between this xylem and phloem, it is called as a conjunctive tissue. So this is called as a conjunctive tissue in between xylem and phloem. This is parenchymatous tissue. It is called as a conjunctive tissue. So this is about the dicot and monocot root. So next one is the differences between this dicot and monocot root. So here we already know that. So here the monocotyledonous roots are fibrous or adventitious roots and which consisting of wide network of thin roots and root fibers, which are originated from the stem. Whereas in monocots, that is in dicotyledonous in dicot root, that is dicotyledonous roots are tap roots and which consist of single primary root from which secondary and tertiary roots will be developed and they grow vertically downwards through the soil. So here the root system in monocots is fibrous or adventitious root system, whereas in dicot roots, that is the tap root system. So primary root, the development of the primary root which stops during the post-embryonic development of the roots, whereas here the primary root continues to grow throughout the life of the plant in the form of the tap root. So epidermal, next one is the difference between the epidermal covering in monocot and dicot roots. So in monocot roots, they are covered by cork cambium after the peeling of the epidermis. Whereas in dicot roots, the covered exodermis, here the epidermis is replaced by the exodermis, which is modified epidermis. So cortex in monocot is, the cortex in monocot roots is wide, whereas the cortex in the dicot root is narrow. And the cortex in monocot roots, which composed of only parenchymatous cells, whereas the cortex of the dicot roots, which is composed of both parenchymatous and sclerenchymatous cells. Next, endodermis. So the endodermis of the monocot roots is thicker, whereas here the endodermis of dicot roots is less thick. And here the Casparian strips are less prominent in monocot roots, as these are only observed in young cells. Here the Casparian strips are more prominent in the dicot roots. Here the passage cells, they are found in the endodermis of the monocot roots, but these passage cells are absent in the endodermis of the dicot roots. Next, pericycle. So in monocot, the pericycle of the monocots only form the lateral roots. Here the pericycle of the dicot roots form the carp cambium and lateral roots. So along with these lateral roots, here the pericycle also forming the carp cambium, because in dicot secondary growth is same. Here another difference in the pericycle, that is it is single layered or double layered, whereas in pericycle pericycle of the dicot roots, it is always single layered. Next one is the cambium, that is cork cambium or vascular cambium, which is absent in monocot roots, whereas both cork cambium and vascular cambium is found in the dicot roots. Next difference in the vascular bundles. So the number of vascular bundles is greater than the 6, that is polyarch condition. Whereas the number of vascular bundles in dicot root is between 2 to 6 only, that is diarch to exarch. Next difference is the xylem vessels in the monocot roots are oval shape, whereas the xylem vessels in the dicot roots are polygonal in shape. Next difference is xylem parenchyma is present. Here, xylem parenchyma is absent in dicot root. So another one is the conjunctive tissue. So the conjunctive tissue of the monocot roots is parenchymatous, that is it is made up of parenchyma, whereas the conjunctive tissue of dicot roots, which is both parenchymatous and sclerenchymatous. So next one is the pith. The pith in the monocot roots is developed and prominent, whereas here in dicot roots it is less developed or reduced. So secondary growth, which is does not takes place in the monocot root, whereas secondary growth takes place in the dicot root. So this is about the internal structure of dicot and monocot root and the differences between dicot and monocot root. Okay. Any doubts?
Okay, my dear friends, regarding of today's class, if you have any doubt, please ask our respected madam. Madam is going to clarify your doubts. So this is wonderful class because of in plant anatomy, the primary structures of the stem, root and leaf is most important. Yesterday, madam has clearly explained about the mono and dicot stem structure, anatomy of primary structure of anatomy of the monocot stem and anatomy of the dicot stem. Madam has clearly explained in previous class. Today, madam has explained about the internal structure of the root of the monocot and dicot. So the major difference between monocot and dicot stems and roots is most important. The difference between epidermis and a difference between the cortex of the mono and dicot, and even though the steel, endodermis, the xylem, the protoxylem and metaxylem, arrangement of the protoxylem and metaxylem, the condition, exarch condition or endarch condition. Okay, so these are the most important points in the internal structure of the stem and root. So madam has clearly explained. Tomorrow we're going to explain, madam is going to explain anatomy of the leaf structure, maybe, or even though the secondary growth. So today, madam has clearly explained, madam. Thank you very much. Regarding of today's class, we are wholeheartedly thanks to you for the clearly explained about the each and every point, and you touch each and every corner point of the primary structure of the mono and dicot root. That is the epidermis, cortex, steel, and in that cortex, the condition, the monozylic or even the polyzylic condition or exarch condition or endarch condition, these are the most important points in the arrangement of the xylem and phloem of the phloem in the steel. So, madam, thank you very much. I will going to wind up the session with your permission.
Okay, thank you, sir.
Right, madam. Right.